Here we are at lesson 3 of chapter 12. So we are going to be looking at DNA replication today. So we'll look at the process and how the structure of DNA allows it to be replicated and replicated correctly every time. All right, so in order to copy the code, we have to have two complementary strands. Okay, so in other words, each strand can be used as a template to make the other strand. So you can kind of see that in the little animation. So what's happening is that the DNA, first of all, that it straightens out. Instead of being a helix, it becomes more of a straightened ladder. And then it unzips, is what we call it, when we break the hydrogen bonds in between all of the base pairs. And then we add new bases according to the base pairing rules. A pairs with T, G pairs with C. And so you end up with two new strands. So instead of having just the one strand, you have two new strands that are each identical to each other and identical to the original. Okay, and that is important because when we pass on our genetic information, we want to pass it on correctly. We want it to be the same as what we have. Okay, so if you think about mitosis, this is how we can end up with two new cells that have the exact same DNA as one another and the exact same DNA as the original parent cell. So this is a not moving diagram showing your two strands. So we have our original strand here in the parent DNA, and then each of these little boxes in the middle is representing the different bases. So you can sort of see the hydrogen bonds in between them. It's kind of blurry, but there's little lines that are representing the hydrogen bonds. So we come through and unzip, or we break those bonds, and then we would make a new strand off of those templates to create two identical copies of the DNA. The replication is controlled and carried out by enzymes. There are several enzymes that are involved. The only one you actually have to know is DNA polymerase. So DNA polymerase, his job is to create the new strand of DNA. And as he goes on and he's adding the new nucleotides, he's also proofreading and making sure he's adding the correct ones. Okay, so we have, let me see if it still works. Oh, it doesn't because it used Flash. That's just sadness. Okay. So I used to have an animation that showed how this whole process works, but with the downfall of Adobe Flash, it no longer works. So that's sad. But here we have just a picture that kind of shows some of the other enzymes that are involved. So you have this enzyme. We'll start here. This green one that's kind of circular, that is topoisomerase, which is responsible for unwinding or kind of uncurling the DNA, taking the helix structure and making it not so helixy. And that lets helicase get in here and actually break the bonds or unzip the DNA. And then here we have a DNA polymerase that's adding the new nucleotides. Okay. On this other side, we have to read it in a specific direction. So one side is called the leading strand and the other is called the lagging strand. I don't think your book really gets into that, but to do the lagging strand, because we have to read from the 3' end to the 5' end of the DNA. To do this, so the leading strand could just like take off and go. Because it's reading in the same direction that the DNA is being broken apart. So just kind of follows along behind the helicase. The lagging strand, though, kind of has to start in several places and create these little fragments. So you have a DNA polymerase that'll just kind of come in and do a little short piece, and then another DNA polymerase will come in and do the short piece behind it. And so you have these pieces that aren't join together, and that would be a problem. So we have another enzyme called ligase that comes and fixes that up. Okay, so it joins those pieces together. But again, I think that part, if you're not really sure about that whole lagging strand and what's happening there, I think that's beyond what your book really covers. The ends of DNA are really important. So the tips of the chromosome, so remember in most animals, we have these linear chromosomes, most eukaryotes, sorry. So everything but DNA, sorry, everything but bacteria. We have these linear chromosomes, so they make this kind of straight line, and the tips are called the telomeres or the telomeres. And it's really difficult for DNA polymerase to get all the way to the end of a strand of DNA because it doesn't have anything to hold on to at the end. And so over time, what could happen is that you would start to lose information. And the telomeres don't necessarily have anything crucially important to the cell. So they don't have like really coding DNA. It's just kind of a tail or a cap or something stuck on the ends to help protect the ends of the DNA. But it still needs to get copied because otherwise you end up with it gets shorter and shorter. And eventually, after several rounds of replication, you would start to lose pieces of DNA. So to prevent that, when we get to the end, when DNA polymerase is done, we have another enzyme called telomerase that comes and attaches to the very end and adds just a little bit of DNA to the telomeres. It doesn't code for anything, but the telomeres are just there to protect the end of the DNA so that we don't lose anything from one replication event to the next. Okay, so the telomeres are really important. Replication does occur a little differently in eukaryotes versus prokaryotes. So prokaryotes, bacterias, have just a single circular DNA, or they tend to have circular DNA. So to copy the bacterial DNA, it starts at one spot. It's called the origin of replication. And then from that spot, it goes in both directions all the way around the whole circle. And then you have two copies of the DNA, and then the cell can go through mitosis. Okay. And so it would be kind of like this. So you have your origin, and you slowly make two new copies until you get to the right. So you're going in both directions. So these areas where the strands are separating is called the replication forks. So you would have two replication forks working their way around until they join back together on the other side. And then you would terminate. They would have made a copy of the whole circle. So they're done. Now you have two copies of your DNA. Each one contains an original strand and a new strand. And we can go through mitosis, and those cells can each have a copy of the DNA. Eukaryotic chromosomes are a little different. So in eukaryotes, remember, one, we have these linear chromosomes, and two, we tend to have many of them. So if we were to start each one in a single place, it would take forever to copy all of our DNA. So instead, our cells will create these replication bubbles. So each one has the two replication forks. So we start replicating at multiple points along the same chromosome. And that lets us copy our DNA so much faster because we're starting in multiple places. Okay, so that's the biggest difference in eukaryotic versus prokaryotic replication is the number of points of origin. Okay. So eukaryotes have multiple points of origin, and prokaryotes just have the one point of origin. All right, and I think that is in fact the end of these notes. Okay, you are doing a lab tomorrow. I don't have that plugged in. Let me see if I can get it to come up really fast. Probably not on this computer. So your lab tomorrow is paper-based, and you are going to model replication. So you're going to get a piece of DNA, and you're going to cut it apart in the middle, and and then you're going to hand you have 2 strands, you're going to hand it to 2 different people in the class, and they're going to try to replicate your DNA. So your paper is going to look like this. Give me just a second. Oh, I don't know if I can pull it up. Maybe any day now it'll work. Sorry, I should have been more prepared. Okay. Now I just have to wait for the PDF to load. Okay. Thankfully, it's a PDF and not a Word document. That would have been sad. I don't have Word on this computer, and it's kind of frustrating sometimes. Da-da-da. Okay, here we go. Here we go. All right, let me share my screen again. Okay, so that's not what you're going to get first. What you're going to get first is this. Okay. So you're going to get a strand of DNA, and you're going to cut it apart right down here in the middle. So let me draw on here. That'll work. All right, so you're going to cut it along this, so you cut it into 2 pieces. Okay, so that might be a challenge for some of you. Okay, so you'd cut it exactly like that. Okay, so you'll have 2 sides, and the directions show you that. We come all the way back up here. They show you that that's where you're cutting. Okay, so you're going to record your original sequence, both sides. Okay, cut it apart. Write your name on the front side where the letters are of both strands. Give it to 2 different classmates, and at the same time, you're going to take one of theirs. So this might take, since there are 5 of you, this might take a little doing to make sure everybody gets 2 different strands that are not their own. Okay. So you are going to then attach the correct nucleotide. So you have a whole bunch of these. These are going to be harder to cut out. You got to cut all the way around, and you got to keep the tabs. Okay. And then you're going to glue them on where they should go. Okay, so you have a tab to glue it onto the piece across, and then a tab to glue it onto the next one on that same side. Okay. So then you do that. You write your name on the half of each one that you made. Write your name on the back, and then give them back to the original owner. And when you get yours back, you record yours here. Here's a hint, they should match this one. Okay, and then you're going to answer these 4 questions. Okay, you need to do that on paper, on your own piece of paper. So you should have this page tomorrow. You will get half of one of these pages, and then you get a nucleotide sheet. Okay, so that's what you should be doing in the lab tomorrow to model replication. Let me know if you have any questions over the process of replication, what you are or are not expected to know. So tonight you need to do the 12.3 homework. It should be pretty straightforward, should be all in your book or in the notes. The notes are posted, or should be. If they're not, let me know and I will get those posted. But I'm pretty sure I tried to schedule everything so that it would post at about the right time.